Welcome to Founders in Biotech. In this podcast, biotech entrepreneurs share their founders' journey and scientific achievements. I'm your host, Sergey Glinka. Today's guest is Marco De Vivo. Marco is the head of molecular modeling and drug discovery lab at the Italian Institute of Technology, IIT. He's also the associate director of the IIT, responsible for computational sciences. His research is focused on development of computational methods to achieve an atomic level comprehension of chemical systems. In the year 2021, he co-founded IAMA Therapeutics. IAMA Therapeutics is developing small molecules for CNS disorders by selectively inhibiting cation-chloride cotransporters to address atypical brain activity in neurological conditions. Thank you so much, Marco, for joining us on Founders in Biotech.
Thank you for inviting me, and I look forward to our discussion.
Well, before we start with science and your biotech journey, first of all, what inspired you to become a scientist? So you studied chemistry in Bologna. Tell us a bit about that.
Okay, yes, that's a long time ago, and that's how I started. Basically, I think I'm very curious in general, and in high school I had a great professor, I think, in science who made me appreciate chemistry. I wasn't very much into the study in general. I had other interests at that time. I was into sport, but in chemistry I found it very interesting because with very few rules, one could create something new. And that to me was already inspiring in a way. And then I actually got very much interested in genetics because to me it was fantastic to find out that only with 4 molecules, 4 letters, one could define then triplets that were defining amino acids that were building proteins. And all this would make a lot of sense from a logical point of view, but it was also elegant in a way, how nature was able to use these rules to create something so magic for us, for life. And I actually wanted to become a medical doctor at the very beginning. And then for some reason, I got into chemistry, and chemistry was close to medicine at that time. It's a long story, but to cut it short, then I decided to start with chemistry and then see how, if it was possible to go towards medicine in a way. And that's how I started. So then I decided to start studying chemistry at the University of Bologna. And the more I was into it, the more I liked it. And towards the end, I was into organic chemistry, but then I wanted to always understand the mechanisms of the reaction that I was studying. And so I found theoretical organic chemistry, which was a way to still remain in a chemistry close to life, to my understanding at that time, even though it's not only related now to organic chemistry, one could say. But for me as a student, organic chemistry was reflecting the ability to go towards pharmaceutical chemistry and drugs. And then, however, with the theoretical organic chemistry, I could start to do computational chemistry and therefore use the computer to study reaction. And at that time, catalysis in proteins, in enzymes. But I always was tricked by the idea of doing something that was interfering then with the diseases and hopefully have an impact in patients.
Yeah, I hear it from time to time with our guests that there is a certain curiosity, but also the fascination for building something. And with chemistry, you can build new things, but also some of them are interested also in having some impact. As you said, you wanted to do medical sciences, and this kind of bridging kind of chemistry with. So you're kind of a doctor by designing molecules. So this combination is very. From time to time, we have this drive, the combination of these three parts.
Yeah.
So you said design of enzymes.
Yeah.
But it's becoming now very with a different flavor with the generative AI methods. But what was it? Maybe briefly, what was it back then? The design of enzymes. What was the process? Maybe we can talk briefly about that because it has changed probably over the time.
Oh, well, actually, the last Nobel Prize was to David Baker
Yeah.
for his Rosetta software, which was at the end of the 90s. So the process, in a way, has become more effective because of the experience now that we have and also the power in terms of computation that we can put into this designing phase, which, can be done before the testing. Of course, there are other approaches like direct evolution. I don't want to get into that,
Hmm.
but the idea is to really use our understanding to pass from function to modulation and therefore designing something that is inspired by nature but artificial in a way. So we can tune the activity of enzymes toward what we want. So we want to change the function, include a new function into an enzyme. So these enzymes are basically proteins. So you can use the scaffold of proteins and then insert at the catalytic site where the reaction happens whatever you need in order to make it more proficient, more flexible in a way, or more resistant to temperature, for example. But this is an activity that I always had because I started with enzymatic catalysis in terms of computation. So actually, I was running quantum simulations to look at how the enzyme is able to move atom by atom its catalytic site in order to break a bond, to form a bond in the native condition. So I'm not, I didn't start from doing new enzymes. I started by understanding pharmaceutical relevant targets for which it is important to understand how they function, because if we understand how the atoms exactly move, then we can design molecules that they can fit into that mechanism and possibly block it usually, or at least modulate this reaction. So that's the beauty for me, to understand really in depth a mechanism. So then we can use this information in terms of drug discovery. Is actually, there is the transition state theory. So
Yeah.
if you have a reaction that passes through a transition state, which is the highest point in energy, and then if you block the enzyme in that state, then the affinity is higher for the compound and therefore is more potent, and therefore the idea is to really understand and then design. Of course, there is a lot of serendipity. You can do high throughput screening. You can start from something and then optimize it. There are many ways to do it. But for me, if you ask me, I got intrigued by the idea of understanding very well something, and then by understanding, passing to the modulation of it with an external probe, a molecule, or in terms of enzyme design, you were saying, is by modifying the enzyme itself by mutagenesis, and therefore drive somehow the mutagenesis to those solutions that have the highest chance to lead to something new.
Yeah. I assume there were proteases involved in those studies for you.
In my case, yes, usually is hydrolysis of bonds. Yes.
Yeah. Yeah. There was a lot of research on
I'm
the
particularly
transition state.
interested since the beginning of my career in enzymes that are able to process nucleic acids, so polymerases, nucleases, topoisomerases. And now recently I've looked at ribozymes, which are RNA-based enzymes that cut RNA. They're very timely and also very promising, also as a target for pharmaceutical intervention. So we are targeting with small molecules, RNA, in collaboration now with a structural biologist, Marco Marcia, who is always my counterpart. I need structures. As a computational person, I need structures because I need models to simulate, and reliable models come from structural data most of the time.
Going back to your curiosity, so you started theoretical organic chemistry, but then you shift to modeling, computational studies. You moved to you did a postdoc at the University of Pennsylvania,
Right.
and then also gained some experience with Rybex Pharmaceuticals.
Right. Yes, that was my path. So basically, since my undergrad student work, I always stayed in computational chemistry, so molecular modeling. Up to then I did my PhD in Italy, and a couple of years in Sissa, in Trieste, and then I went to Philadelphia, University of Pennsylvania with Michael Klein, and that was in the physical chemistry department. So it was very much oriented into physical chemistry of, and in my case, enzymatic processes. And I was using high costly simulations at the quantum level to understand enzymes. Now, this was to me super interesting, and it is still today. But at the end of my postdoc, which was a fantastic experience because I had the opportunity to join a top level lab with top colleagues that at that time were postdocs with me. And so you're surrounded by these talented people who push you to give really the best of you and to grow. So it was a fantastic experience for me. But then I really wanted to do drug discovery. That was my dream since the beginning of my studies in chemistry. So then I decided that I wanted to have an experience in a more practical environment, in a sense, not only physical chemistry with quantum simulation. So I decided to apply to a job at that time in New Haven at Yale. It was a spin-off of Yale. So it was funded by Bill Jorgensen, a pioneer of computational methods in drug discovery. Thomas Tides at that time, Nobel laureate for the crystal structure of the ribosome. So I could join a fantastic, scientifically fantastic environment where I could work on crystal structure of the ribosome, which at that time I didn't think of it in this way, but it was mostly RNA. The ribosome is 70% RNA. I was actually targeting part of the structure was made of RNA. But in that environment that I joined, I was leading a group where I had medicinal chemistry now, pharmacologist, structural biologist in the same team. And I was coming from an experience where I was working as a postdoc in simulation with supercomputer and at the quantum level. So it was totally different for me. So when I started, it was really another experience where I really had to design and synthesize and test compounds to target the ribosome as a new antibiotics. That was the target of RIBEX. So RIB stands for ribosome. At that time, X, because there were X-ray of the ribosome. Mm-hmm. And every couple of months, we had new structures of the ribosome. It was a great experience for me where I could step up from the level I was before doing only computation, now dealing instead with the medicinal chemistry and all the aspects, even PK, pharmacokinetics and everything. And I was only there for a couple of years. But at that age, in that environment, which was quite intense because every week we were on the verge of a big discovery, and then, oh no, bad results, and then, oh yes, we're going to make it. So, and there was a very limited number of people involved, but I could also attend to the group meetings of Bill Jorgensen in his group at Yale. So there was still a sort of academic environment for me. I could realize that this is what I wanted. I wanted a group like that, ultimately, where I could do science, intellectually rewarding, understanding the mechanism, but then design compounds as new drugs or drug candidates to become new drugs, hopefully in the future. And that is what gave me the understanding with myself of what I want, what I liked. So after a couple of years there, by chance, because I wasn't planning anything, I was going with the flow in my career. The Italian Institute of Technology was founded in Italy, and the positions were available, and therefore I was through contacts. I got to know that there was this new institute in Italy, and it was potentially a very good option for me in the future to start my own group. It was a tough decision because I was in the States at that moment.
Yeah, it sounds really tough because you said you were a couple of years very involved in this. Focused, you have the team that run, there are ups and downs, and it sounds like there was progress, but then you kind of switched to academia, right? And my question would be, in this context, when you got this invitation, was there a flavor of this drug discovery option already there, or was it your idea to use this experience that you had and gradually build it into in the Institute of Technology?
No, there was the idea to have a group focused on drug discovery ultimately, but starting from computation, which is what really at that point I wanted. I was very happy and very satisfied with my experience in RIBEX. On the other hand, I must say I was feeling the urge to be independent in a way, to have my own projects, my own initiative, and that, of course, was not possible in an environment like that because there are priorities.
Not unlimited resources. There's focus, and of course,
right. So I had I think I felt I had learned what I could I also gave back what I could at that point, but there were also other factors, because it was a change in my life that was quite drastic. For after more than 5 years in the States, I was going back to my own country. I felt it was a train to catch. Who knows if it's gonna be another good one like that in the future? And also, I wanted to have this more academic profile, and after 2 years there, I felt that maybe then it's going to be too long if I stay away from academia for longer than this. So there were a combinations of factor, and at the end I decided, yes, let me try and go back. But
Yeah,
it
I
was
understand. Like if you want to be independent, that you kind of lose this skill or the urge for the ideas. I understand. And there was many years in like you spent leading the group and building the methods and publishing. And in 2021, probably there was data before 2021, maybe. So you decided to start or co-founded IAMA Therapeutics. There are probably two things. First of all, what led that to your personal, okay, I'm going to start a biotech in parallel to the academic research? And what kind of data was convincing? I mean, you have those beautiful papers that show, I will talk about in a minute. What was the thought process maybe for you, or the process to start a company?
Right. Yeah. So this is something that I had in the back of my mind since the beginning, as I said, because it was, in a way, the best scenario for me, because I would have had my freedom and my group doing science. On the other hand, I was an entrepreneur with something that I would help me push my science to the clinic. And that's the only way to do it, unless you in-license the compound to somebody else. So the company was something that I had in mind. And I was fortunate because years before, I had started a project within IIT with a colleague who is a top neuroscientist. Her name is Laura Cancedda in IIT, and she was the expert of the target. And that actually made everything easier in a way, because then I could start collaborate with a scientist in my own institute, actually door to door. So everything was easy in a way, also in terms of contacts, initial collaboration. She had published a paper a few years before on a mechanism that was a promising, and a protein that was a promising target to treat neurodevelopmental disorders. And the story was actually pretty easy. So basically, she demonstrated together with others that there is an FDA approved drug, which is a diuretic and is called bumetanide, which was, however, able, if you reposition the drug from being a diuretic to be a drug for CNS, to act in neurons and restore some cognitive problems in Down syndrome models, in animal models. So you have a drug now that, if you look actually at the effect that it does in the brain, it acts with a beneficial effect on CNS. The problem is the fact that it's still a diuretic drug, because it acts in the isoform in the kidney.
Yeah.
So the target in the brain is called NKCC1, but the isoform in the kidney, which is the primary target of this drug, is NKCC2. So now the diuresis becomes a side effect if you want to reposition the drug for the brain. So when I met her and we started to discuss about projects and the activities that we were doing, I say, well, maybe there was also an input from an internal advisor at IIT, was connecting the dots between what I was doing and what she was doing. And they say, well, why don't you try to develop a specific drug for the brain, so we don't have anymore the side effect on the kidney? And that was the entire rationale at the beginning of the project. Then for more than 4 or 5 years, we worked together, joining our efforts, where I am the chemist. So I was taking care of all the designing, synthesis of the process, and she is and was the neuropharmacologist. So she had all the in vitro assays, she had all the in cells up to animal models. So we could, at the beginning, it was quite difficult, I can tell you, because there was no knowledge of the structure of the receptor. In this case, it's actually a transporter in neurons, the one that you introduced, the NKCC1 ion cotransporter in neurons. So we had to start with what we knew at that time, which were diuretic drugs that were actually affecting unspecifically, so they were acting on both the protein in the kidney and the protein in the neurons, the function of this protein. And then we started to screen many compounds, internal libraries. We built a pharmacophoric model based on diuretic drugs that were aspecific. And then the project started. Till, if you want to get to the end, the fact that over the last few years, the resolution of the protein NKCC1 was resolved. So we had access to the crystal structure, the cryo-EM structure of NKCC1. That was a game changer because, of course, then you put everything together. Now the information that you have in the context of a structure, and structure-based drug design works very well with the methods that we have nowadays. I can go in detail in that. Yeah. And then we had a new chemical class, so new chemical entities that we could patent. And we patented these compounds with the data we had at the time, including efficacy in animals. So we had optimized this new chemical class up to efficacy in animal models. And that was the trigger. At that point, we had a technology that was ready to be taken into advanced preclinical studies. And so together we decided to look for investors. And actually, we started a process because it wasn't like this that we found investors, especially in CNS, as we can discuss later.
Yeah.
So it took almost 3 years for us to present the project to many investors, learn also how to present because we were scientists at that time, not entrepreneurs, and sell the idea, convince the investors till you find somebody with whom you have a click, where you share the vision, they like the project, they like you, you like them, and therefore then we started the company.
There are not many, also like what I learned, the landscape of investors who are looking to CNS is, so not every biotech investor is looking to CNS. There must be specialized. So let's dive into CNS drug discovery. I think, and I will look at your at papers that are here just to have some, and we will share it also with the audience. So what is striking is that in the last decades we haven't seen breakthroughs in disordered for CNS drugs. Also new indications. So in addition to the typical properties that CNS drugs must have, what is the barrier that in your experience? Is it because you said that Laura had the models there, right? So what is because if you say it's decades, no breakthroughs, what is the actual barrier in CNS drug discovery, independent from the drug properties? Let's treat it as separate, multi-parameter optimization problem.
Right. So the problem is, of course, the perceived and actually to some extent also verified high risk of failure. So investors do not like to take too much risk. They want to de-risk the project. So, and of course, in CNS, the big issue is the poor or the limited, you can say, transferability of efficacy from what you have in animal models to what then it is discovered in phase 2 clinical trials. And this is a risk that unfortunately is inherently contained in the fact that we're dealing with the brain. So if we develop an antibiotics, of course, the bacteria that we kill in mice most likely respond in a similar way in a human body. But when we deal with the complexity of mechanisms that are regulating diseases in the brain that are by definition multifactorial, because if you take autism, it's a spectrum disorder with many possibly mechanisms involved in it, and or even Alzheimer's and so on, we want to reduce it to a single target that actually is affecting the disease. And of course, we are talking disease. For instance, if we take the disease we go after at the moment the most, which is autism, is not about curing the disease, first of all, because it's not possible. Okay, so you treat the symptomatic effect of the condition. It comes down to populations that can be treated with a certain drug rather than another, and therefore it's key patient stratification. It's key to identify responders. It's key to identify biomarkers in order to have a better stratifications of the populations that you want to treat. All these aspects are critical for CNS drug discovery because it's so difficult to be successful when you go and measure efficacy. And this is, again, due to the complexity of the disease. So we have to somehow, if possible, either reduce the risk or the perceived risk associated with CNS, or accept that CNS will always, for some time in the future, have a lower percentage of success compared to other areas of drug development, just because it is more difficult. So the transferability of the efficacy is the challenge. For what we can do in animal models, which is not my science, I think it's totally different than mine. But since you are correct, we can divide the challenge in terms of chemistry, and I don't think that is a challenge that honestly worries me that much. I mean, we have always to say, well, but then we have to penetrate the blood-brain barrier. Okay, it's a challenge, but we know how to solve this problem. And by the way, it's not that we need a great amount of drug in the brain. It may be enough just a little bit to have target engagement and see efficacy. So that part, to me, it's okay. I can deal with that. It's challenging, but we have learned over the years how to at least approach that part of the problem. The other part of the problem is very complex. How to model such a complex disease in animals and then transfer these into a clinical settings. Now, this is, in fact, if I can add on this, one of the strengths of our proposal to investors at the very beginning, because the drug that acts on NKCC1 in the brain, but also on NKCC2 in the kidney, so this bumetanide diuretic, has been taken into clinical studies and has shown activity in phase 2. Then we can talk about phase 3, but phase 2, it was okay. So at that point, you have a mechanism that has been validated by a drug in clinical settings. So you know that if you block that protein, there should be the same transferability at that point, because it has been seen to be active in clinical settings. And actually, it failed in one single clinical trial in phase 3, but they had to lower the dose compared to phase 2 because they had too much dropout of patients because of the diuretic effect. So now there is an issue for phase 3. They had to go with lower dose. And that this could be actually one more motivation to have a selective NKCC1 inhibitor, because you get rid of the diuretic effect, which was actually a huge problem in phase 2, even though it was working.
Okay, understood.
So that was a de-risking factor. And so the more you lower the risk for investors, the better, of course, for the project.
So the big talks, like red flag was not there, like systemic talks. If you, at least for this type of compounds and also for this mechanism, has been solved by the clinical trial. But that's very interesting. In addition to the optimization towards drug-like properties, I have 4 points here. Like I like this perceived versus verified risk. So there is a kind of it sounds like there are question marks, right, that we hesitate to address, right? And then there is transferability of animal models to phase 2 data, like humans, which makes a lot of sense. Clinical strategy, probably also population size is doesn't, like, if it's not large enough, it's not attractive. If the list of risks for transferability is high. So that was really helpful to have those points, I think, because they're generally not discussed in in CNS drug discovery, at least from a non-CNS insider. And like you touched upon this selectivity of subtypes. So you started with a compound that's already known. You've run several computational studies to build a pharmacophore model. Can you guide us through this? How you in details how you approach this problem. First of all, to find FISCM similar compounds versus how to gain selectivity over like I need to look exactly NKCC1 and NKCC2, right?
Exactly. Yes. Yes. It's the one is in the kidney, the two is in the kidney, the one is also in neurons. So then you want to go only on neurons at that point. So the first, as usual, that's my approach, but I think I can make it general.
Oh, let's Marco, let's pause for a second. Let's like this. What is the role? Let's dive a bit into physiology of this receptor. Generally, what is the role of this transporter in the physiologic context, and then pivot to like the selectivity among the receptors.
Right. So in neurons, this transporter allows the entering of ions. So at every cycle, there are two chlorine ions, one potassium and one sodium ions that they pass through the transporter that are transported inside. So NKCC1 let these ions go into the neuron, and this controls basically the concentration of chlorine ion in the neuron. The concentration of chlorine ion then is related to the GABA transmission, excitatory inhibitory, and therefore to the effect that you have at systematic level. The point is therefore that has been shown that too much chlorine concentration in neurons is related to symptoms in neurodevelopmental disorders. Mm-hmm. So what we wanted is to block NK, so to restore the correct concentration in neurons of chlorine ions. The same happens in the kidney with ions, but in that case you produce the water and therefore you have diuresis. That's another mechanism. But the concept is always that there are ions from the outside that enter into the inside of the cell. So in neurons, there is this cotransporter that let ions get inside. Rational for us was, which is what happens in fact with bumetanide, is that it blocks the transporter, therefore chlorine cannot enter anymore, and therefore there is you restore the correct chlorine concentration into neurons. That's the physiological mechanism that happens. So at the beginning we had a compound that was able to block both NKCC1 in neuron and NKCC2 in the kidney. So then we started to modify the compound to understand what were the chemical features that were necessary only to block NKCC2, which could be therefore eliminated or changed in a new scaffold to retain activity only on NKCC1. And this was a trial and error. More elegantly, I can call it structure activity relationship. Yeah. That is done over the years, trying to understand exactly what were the structural and physicochemical properties of the scaffold that we were starting from to give some sort of selectivity. So we actually worked initially a lot on bumetanide itself, because we started to do kind of reverse engineering. So let's change bumetanide as well. Let's see if we lose one of the two targets, and therefore we become selective, or what it takes to become selective. And then we transferred what we learned on a new chemical scaffold, which was anyway inspired, if you want, by bumetanide, but that was taking only into considerations those features that in bumetanide were giving us some sort of preference, because then we wanted to really shift the preference towards NKCC1, which was our target. And that's what happened over a number of cycles, because then it's really a loop that has to continue, where you make compounds, read out the result, and then you adjust your hypothesis, you do new compounds, and then you go in this loop till you find what you want.
So it was initially kind of ligand-based SER building, but
initially, because we didn't have the structure of a KCC1, but then we found something that was promising in terms of chemical structure, that was giving us some selectivity, and then it became even more, in a way, efficient this process, because, as I said, NKCC1 was resolved, so we had access to the structure. And that is really a plus in terms of designing, because now we knew the space. We knew exactly where it was fitting. The selectivity, however, remains challenging, I have to tell you, because the selectivity can come from the fitting into the structure, but it could also come from a different way the compound exploits to reach the target. So that is really something that builds after potency. So first of all, we want to be potent and new, a new chemical scaffold that was able to target NKCC1, and let's hope for the best regarding the rest. And then we started to optimize the structure, only looking at off targets, including NKCC2, which at that point for us was an off target, something that we didn't want to touch.
Yeah. So you did first the SER, then pharmacophores, then this structure was probably released, right? And it sounds like you could gain some selectivity because of the different compartments in the body, right? So I'm not sure if it's like the chemistry is easy, but if you could, trap it in like after passing the blood-brain barrier, that could provide some selectivity. Is it possible? The
Difficult to
PK, I
say
mean
in
the
this
PK
case,
parameters. Yeah.
because
Okay.
anyway, even if it doesn't pass through the brain, you still get to NKCC2, which is outside the brain. So the first target that will be reached anyway is NKCC2. No, I think it was really, so there are parts of the compounds that we found quite critical for activity, and in fact we couldn't touch, basically. So that the moment you touch those chemical part of the molecule, you have a detrimental effect. There are other parts where we had more flexibility in terms of groups that we could explore. And in those areas, in those positions of the scaffold, we really explore diversity in terms of chemistry, building a structural activity relationship that was actually not activity, but selectivity in that sense. So we wanted to be selective. And that led us, so because in terms of, I didn't say this, in terms of potency, for the design nowadays, we mostly use free energy perturbation calculations. So this is a method that is nowadays like the gold standard for designing compounds once you know where they bind and once you are in the lead optimization phase, which means small changes to really increase potency. And that works, I think, beautifully. And this goes back to the FEP theory and the setup. In fact, most companies nowadays, if they find themselves in a situation where they have a target and they know where the pocket is and how the compound binds, FEP is very effective. But selectivity is really basically binding to another target, which is very similar. So it could be difficult to rationalize these small changes.
But do you, like, follow-up question, do you use FEP also to optimize selectivity? So if you see like some changes provide potency there, but we lose
We are trying, but the pocket itself doesn't lead us to clear indications how to exploit the differences. So it's more about, there are a couple of places where we can play with different flavors of substitutions. And in those sites, we try to see trends in terms of selectivity, and then we build according to that, retaining activity, because activity comes from the rest of the compound, which it fits well in the cavity. And we know that because we have data demonstrating the binding in the pocket. So
going back to your, I think that's the Jack's paper is really interesting because you were studying mechanistically, like the mechanism of enzymes, right? And now you are, I think, have this huge movement. It's also mechanistic movement, how ions are transported. My question will be, and there are like 12 transmembrane helices, and there is also the membrane. Have you simulated all that? Was the for me, it's the question, was the mechanism obvious when you simulated long enough, or was there some bias to get to this model that you have published?
Okay, so that's this goes back to how I wanted my group and why I got interested in chemistry at the very beginning. So I really want to understand the details of how these beautiful machines work, no? So once I could block it, but I really wanted to understand what I was blocking and therefore use this information to block it even better. So simulations of this type are very, very useful because you see the time evolution of atoms over, of the system and therefore the mechanism. So this is a complex system, as you said, because it is a protein embedded in a bilayer, so the membrane with water on top and bottom, extracellular, intracellular. And now you want to see also the ions that pass through that protein and are
for a
able.
parameterization, I was looking at it. Parameterization, you need to be very good at it.
Well, but nowadays for experts in the field, there are ways to do that, and parameters can be chosen, and there are parameters for basically everything. The challenge is not in the parameterization. The challenge is in the what is called the sampling of the configurational space, which means how many conformations can we really explore and therefore investigate in terms of mechanisms. How do these conformations affect the final mechanisms that I'm gonna test in these simulations? And you have to consider that now we are talking about a transporter that has to do this somehow, okay? And this motion happens in time scales that are way longer than what we can explore nowadays. Even with supercomputers, we are not able to run over, let's say, tens, hundreds of microseconds, maybe a millisecond.
So it's a microsecond. Or like, what is the range for this?
The range for the simulations in the tens, hundreds of microseconds normally. Now you can even push
but it's
it
a
a
large
bit
system. It's not just a protein in solution.
It's a big system, but these conformational changes take more time than what we can explore with what is called equilibrium molecular dynamics. So we don't do anything. We let the system run according to the law of physics, and let's see what happens. So then we need to accelerate these events. Otherwise, we cannot see them. So this is called enhanced sampling because we speed up the process so that we can observe it. Otherwise, it would take too long for us to observe it. And to do so, there are many different techniques that can be used to accelerate the process. But if you accelerate the process, somehow you are biasing the process because you are telling the process where to go, where to happen. So in a way, the challenge is bias the system in the least you can, first of all, but also in the correct way. And how do you validate the correct way or not? Well, you look at the result and then you compare it with experiments if it makes sense or not, what you have observed.
Okay.
So to see the motion of the protein that we are describing, it took, this was a work of a fantastic PhD student. Now he's graduated, Manuel, the first author of the paper, who spent the entire PhD working on these simulations. And the main challenge wasn't to set up the system, but it was to speed up the process, so to see the correct motion. And what is the correct motion? The correct motion is the one that allows the ions to pass through and then match the experimental data, reproduce the structures, reproduce the value from experiments that we can, for example, use to validate our hypothesis. There are mutagenesis experiments that have been driven by these results, which validate the simulations themselves. So it's a very complex process that, however, was very intriguing because there are possible mechanisms that are reported for such transporters. There is more than one. So the thing is, which one is okay for, which one is the one that is used by NKCC1 to operate? And the answer was, in this case, the rocking bundle mechanism, which is something like this. So only one part of the protein moves and the other one is stick, and then you see the atom passing by.
Is it
But this is very useful. Also, I'm going to conclude, also for the drug discovery part, because then we know what are the critical residues in the proteins that are needed in order to have this motion.
Yeah, I was like, is it a passive mechanism, like passive transport? There's no active.
There is an action. So the ion diffuse inside. There is no gradient in our simulations. It's not like an ion channel, but there is a motion of the protein. So the motion is needed in order for the ions to go through.
But it's not accompanied with some hydrolysis of cofactors.
So if you now take a bigger picture than what we can run, there are other parts of the protein where there is bound ATP, and the ATP hydrolysis could be coupled to the motion. Yes, we were not considering the source of energy for the motion itself, which was again biased in a way. So we were accelerating this process in order to understand how it was happening.
Okay,
And
the
this is
mechanism.
actually possible. So it was
But
possible because at the end we had to give up the normal conventional methods, and we could use, thanks to the collaboration with Michele Parinello in IIT, one of the pioneers in enhanced sampling methods, machine learning guided simulations. So machine learning in general, are based on ingredients, and the most important one are data.
Mhm.
So then we collected a lot of simulations in different states. We wanted to go from A to B. So we had a lot of simulations of A in different conformation, and then another state, which is B, in different conformations. And then the machine learning algorithm could suggest us a way in the space. So this is it's called a reaction coordinate. So a collection of degrees of freedom that was able to lead the system from A to B. And this is very useful because then you follow this tunnel, if you want to go from A to B, and therefore you can reach the other state with a line, imaginary line you can think of. But the collection of degrees of freedom are ways to describe the systems was retrieved through a machine learning algorithm.
I assume that this model can be used to design molecules. Like if you have different states, you can design like backup series that target a different state.
Yes, absolutely. This is what we have actually done in IAMA Therapeutics after we have nominated our clinical candidate that has now ended phase 1. Now we have additional classes of compounds that have been developed through the information also retrieved from these simulations. It's not only per se the compound, it's more about conformations that are functionally relevant, because you can have the protein in different conformations, of course, and not all of them are going to be critical for function. Of course, if you block, the mechanism just at the moment when it is needed to pass there in order to function, then it is easier to block it. So there are certain residues that we want to engage. There are certain conformations that are more relevant than others. For example, the protein can be open on the outside or open in the inside.
Yeah.
So now it is known that the pharmacologically relevant conformation, this is already reported, is the one that is open in the outside. So there is no advantage in targeting the inward open conformation of the protein, for example.
So coming back to the multi-parameter optimization of compounds, how extensive was this optimization? Like the biology is not solved, but you had the models to measure that. How because I just want to touch this point when going from academia to a drug, this stage with pharmacokinetic, pharmacodynamic, selectivity, unspecific tox, etc. How would you describe with IAMA Therapeutics? How was this process? Was it straightforward because you had worked for 5 years on that
Right.
and built this SAR model? You knew which functional groups, etc. Or did it require additional like optimization of lead molecules prior to nomination?
Okay, so the campaign inside my group, together with my collaborator, was for us was expensive in the sense that it took years and many compounds. If we talk about an academic lab, if we were Pfizer or a big pharma, would be nothing, because we have nominated basically a clinical candidate with something over 100 compounds.
Wow, that's great, actually.
Yeah, exactly. That's great. Still, given that you employ students and or postdocs to do so, it takes a long time and is not the same context as a big farm. But it was very efficient, and therefore, I think there are several ingredients for this result. First, I think we can allow ourselves to be really rationally guided by it. So I always want an hypothesis for the compounds that we do these compounds. Maybe we spend more time on the hypothesis than others, but I think you really can increase the chances to do something that makes sense rather than make compounds because I can, because there are reagents, because there are 30 chemists working on it, and let's explore everything and let's see what we find. I cannot afford that. So we need to be very careful in the way we design and prioritize compounds. The other thing that I find interesting in an academic environment is that we can take risks which are not typical for, I think, for big pharma. Like big pharma go after conventional targets. They want to the risk, their activities. And then there is us doing these very catchy, maybe top-notch research in an area that who knows if it's going to lead something. It's easier for them to let us doing this science and then maybe buy us if something good comes out. So we have the luxury to do stuff that is not meant necessarily to lead to a clinical candidate, and this gives us freedom to think outside the box and do stuff that is not really what you would do in a conventional drug discovery setting. And this is one plus, I think, of doing drug discovery in your own lab. Then when we enter into discussion, we had already quite extensive characterization of our leads compound, which were a few that were for some reason okay on one side and maybe better optimized on another, and there was another one, and then we entered into the discussion with this pool of few compounds, and then eventually in the first work through the advanced preclinical studies it was clear which one was the most promising one, and the backup ones were there just in case. But however, to convince the investors, as I said, it was first of all the de-risking factor to have a clinical candidate ahead of us on the same mechanism that however was not good in terms of developability because of the diuretic effect. It was the criterion number one, to have a de-risk factor for us. And then you really need to characterize the compound more than what you would do just for the paper. Okay, so you need to go also through off-target panels, characterization that usually is done through a CRO. In that sense, the Italian Institute of Technology allowed me to have some work done that goes beyond what would be typically possible in an academic setting. We have facilities, for example, within IIT that can measure metabolic stability, solubility in different, all stuff that is more usual in a biotech setup rather than an academic setup. And this, however, enriches a lot the package when you present your product to investors, because again, every experiment is one step more towards de-risking the project, and therefore there is always one experiment missing according to them, because there is always something more that you could do, but at some point you have to find an agreement,
the right balance. Risk profile. So what, like based on your experience that you said, there is an ecosystem for testing molecules, and I think coupling it with limited resources and better modeling is a really nice combination. And how do you manage those activities? Question number one. Maybe question number two, what would you advise academics to think about before they start their biotech journey, if they are considering it? And would you recommend maybe to, going like focus on the biotech or something? I mean, running those roles in parallel, can imagine it's quite, you need time management for that.
So how to manage my double hat, it's challenging, and honestly, I just think that it's about being passionate about what I do. I cannot give up basically anything that I like. So I like my freedom with my group and young students and talented, young scientists that I enjoy leading and hopefully inspire. And therefore that part, I really love it. On the other hand, I really love the idea at this stage of my path to have an impact on patients, which is what I really wanted. And therefore, I think there is no other way for me rather than passing through a biotech, because at least I'm still heard, influential in a way in what happens in IAMA, even though I'm just a founder and an advisor, but I'm still very much involved. So it takes a lot of time, to be honest. I mean, there is no shortcut here. It's not that I give up something for something else. I just try to keep up with everything and do my best. So my suggestion for those that are interested in doing this is there is a practical side of this, which is to have a project that is really advanced with the idea to be presented to investors one day. For example, it is very important to have a patent. And if you don't take this into consideration when you publish, or anyway, when you make the patent, which is also important because you want to have possibly a strong patent, it's going to affect your appeal with investors. So that's very, very important thing from a practical point of view. And the other aspect instead is more related to what I said before, is that it's going to take a lot of your energy. It's not something that you say, well, but I'll do it in my spare time. I'll found a company. Let's see what happens. If you do it in that way, most likely, I hope the best for everybody, but most likely it's not going to work because it's going to really take a lot of energy. Even just the fundraising phase, it is an exercise that is new to scientists, where you have to deal with different people, with a different language, with different mechanisms. I sat at tables where there were these investors that discussed things that were not familiar to me: pre-money, post-money, business plan, all these terms. You have to become acquainted to these terms, and it takes time. So you need to be passionate about it, otherwise it's going to be a struggle.
Yeah, that's a great advice. Yeah, it's gonna be hard, so you better be passionate about it. Was your experience with Rybex helpful, like with maybe before starting IAMA, or the company is running? Is it kind of, do you have some experience that is helpful for you?
It was extremely helpful for me in this sense. This is where I learned from my supervisor at that time, and also the environment per se, how to manage a group that is a multidisciplinary group in nature. Okay, so because I started my own group as a computational science group only, but then now I have half of the group are experimentalists. I have medchem, I have biochem, of course I have compchem, but even compchem is divided into those that are more dedicated to applications in drug discovery, and actually those that are running and developing methods to study how atoms move. So it's totally different. And I've had this experience in Rybex, where I was actually discussing with the medchems what were the priorities and how they think, how they deal with these problems. It was super useful for me on how to set up my group. Not really with IAMA per se. IAMA is the result of that. But the way I envision my group and the way I run my group in a way is very much as I've seen it and I've done it in Rybex. So it was very, very useful.
That's so great to hear that. It kind of echoed into the setup of the company. And I mean, you must be very patient, because it took, 17 years since you started your group? Well, maybe 15 years, if we go back 15 years. So you can translate the research into new CNS drugs. So maybe last question, what would be your wish for future of CNS drug discovery, very broadly?
Very broadly, is that we increase the rate of success by possibly investing more into it, because I think we cannot be scared by the failures. We have to be inspired by the few successes that hopefully will happen in the future. But the few successes will happen if we keep investing in it and not be scared of it. There is a component that we cannot address. The brain is complex. We don't know much about all these difficult multifactorial mechanisms related to diseases, and I think we have to live with the fact that it's more complex. So you can be scared because of this complexity, or you can be motivated by it. And I understand there is a lot of money into it, but think about the outcome. We are talking about an untapped potential in CNS. There are many diseases for which there is no treatment at all. So if we break the barrier there, the return will be huge, and patients will be super, or individuals, sometimes we have to call them, for some of the indications. It's huge what you can achieve. So more broadly, I think we have to be more optimistic about it and see what happens. I think science will advance. We will learn more about the mechanisms, therefore more targets, therefore hopefully more clinical candidates, and hopefully blockbusters in the future in CNS that will motivate investors.
That's what I think. What you said just resonated strongly with me. Being motivated by complexity and not scared. Like, complexity should like it can be scary, but being motivated by complexity, this is very, very inspiring. So thank you, Marco, for joining us on Founders in Biotech today.
Thank you, Sergey, for this time together, and I'll keep you posted.